Well, there it is, the protein folding problem. Why haven't we solved it? Well, people argue that it's too complicated, and so many people now say, well, it's passé, the problem is passé, but that sounds like sour grapes to me. More likely, it's we haven't solved it because we are missing something very fundamental. And what would that be? Well, the protein doesn't fold in a continuous, homogeneous medium. It falls in water. Water is a very resilient solvent that doesn't really give up its local structure easily. So it's a liquid, but locally it's structured in a very stiff manner. And the protein, as it falls, tends to disrupt the structure of the solvent. And that comes at a cost. The cost can be cast in thermodynamic terms, and it's not small. In fact, I have calculated recently that the interfacial free energy, that is to say, the reversible work needed to span the protein-water interface, is actually extremely significant, and so proteins tend to minimize this reversible work as they fold, as they seek for a conformation that minimizes their intramolecular energy. So this interfacial contribution has to be taken into account. And of course, implicit solvent models haven't incorporated it, but even explicit solvent models haven't properly incorporated it. And let me tell you why that's the case. The prevailing paradigm in regards to how to treat electrostatic interactions in water asserts that the Debye ansatz prevails. In other words, water polarizes in a way that aligns with the field imposed by the charges of the protein. But that's not actually true, especially if water is confined at the interface in cavities that have nanoscale dimensions, right? In the sub-nanometer cavities, water behaves very differently from the bulk, and especially from the kind of context that Debye was envisioning in 1929, when the structure of water wasn't even known. So what actually happens is that water is confined partially at the interface, and so it's unable to align with the field imposed by the protein. And so this is not a Debye term. This is actually an anomaly. This is an anomalous polarization, or I should rather say orthogonal polarization. In other words, it has a component that is orthogonal to the projection of the polarization along the direction of the field. And so there's an energy stored by this quote-unquote anomalous polarization. And that energy actually translates into the interfacial free energy, which is the reversible work that needs to be performed to span the protein-water interface. So what one is actually minimizing is a much more complicated quantity than what's originally believed. But that means that somehow one would have to incorporate this resilience of water, this stubbornness to preserve the local structure of the solvent, in the proper treatment of the protein folding problem. So, in a sense, one would say that the matrix has to be perturbed minimally throughout the folding process. And so you can imagine that there may be an action principle at work, but more significantly, there's a minimal episteric perturbation of the matrix, of the water matrix, that can be translated in terms of an elastic term, an elastic integral, where you contribute to the integral every time that a water molecule loses some of its hydrogen bonding partnerships or departs from its tetrahedral ideal lattice structure. So when this is properly incorporated, you can see that why has there been so little success in terms of the protein folding problem. And one reason is that it's not that really that conformations cannot be found and predicted to be suitable minima in terms of intramolecular free energy. The problem is that those conformations are at odds with the solvent. And to be at odds with the solvent essentially means that the conformations are actually perturbing the matrix too much in a way that would actually introduce a very significant interfacial free energy. And so those conformations are going to be subject to a significant instability, would be destabilized because they are too disruptive of their surrounding medium. So you could compute that in two ways. None of them comes cheap. One of them would be to simply evaluate the elastic energy integral, which, as I said, would try to restore the tetrahedrality of the water matrix, or it would actually compute all the departures from bulk-like structure in the water matrix determined by episteric perturbations. Or alternatively, you could compute the electrostatic energy associated with the breakdown of the Debye approximation. That seems to be a more promising and simpler way of dealing with it. And it shows, I mean, this approach has some very promising features in it. One of them is that, first of all, one needs to admit that the Debye approximation breaks down when there's a nanoscale confinement of water. Now, Debye is nowhere to be blamed for that because he lived in the early part of the 20th century when the structure of water was not known. Linus Pauling hadn't really yet described the structure of water in terms of hydrogen bonds, hydrogen bonding partnerships. So Debye is not to be blamed for not taking into account possible orientations or that would produce a polarization term orthogonal to the external electrostatic field. I mean, you could have never guessed something like that if you didn't know what the structure of water was. However, current progress in the field makes it almost inexcusable for people not to assume that that there is a breakdown of the Debye polarization ansatz operating at the interface and producing electrostatic energy stored at the interface. In a sense, you would say that the protein folds to try to approach the Debye limit. In other words, the protein is at odds with the solvent. Well, initially it's not at odds with the solvent because all the polar groups are properly solvated. Now, the polar groups cannot be buried unless they are engaged in pairwise interactions. So, that's the other side of the coin in regards to the hydrophobic collapse. So once the chain is collapsing into a three-dimensional structure that can be sustained in water, the water is also perturbed and it has a significant non-Debye polarization, which needs to be minimized because the non-Debye polarization is indicative of a significant perturbation of the water matrix. So one would argue that the restoration of the structure of water in the course of the protein folding process speaks of an action principle whereby the folding process tries to minimize the disruption of the matrix, and that is equivalent to saying that the protein folds to approach the Debye electrostatic limit, because try to imagine what would be a protein conformation that would impose a minimal disruption of the water matrix. Well, that would be one in which water behaves almost like in bulk. But if water behaves in bulk, then its polarization aligns with the electrostatic field imposed by the protein. So that is actually the Debye scenario. So you would say, in essence, that you could actually look at proteins and infer something very significant about their reactivity or their capacity or propensity to engage in associations by looking at how much they depart from the Debye assumption. And you can quantify that by integrating the electrostatic energy stored in the orthogonal polarization, in the polarization component, which is orthogonal to the Debye polarization. Or alternatively, by computing in an elastic term all the disruption of the water matrix, viewed as an elastic contribution. Now, I keep saying that, but I never said how are these two different ways of computing the interfacial free energy, how could they be related to each other? And that comes quite naturally if one can actually introduce these ansatz whereby the orthogonal polarization is proportional to the gradient of a quantity that I name G, which actually represents the average number of partners of a water molecule at a particular position in space. So if G changes from 4 to anything smaller than 4, well, that gradient is commensurate with the orthogonal polarization, with the non-divide polarization. And so one could actually solve the protein folding problem taking into account the significant disruption of the water matrix that is brought about by the folding process. So you could, in principle, say that the protein folding is actually an interfacial tension problem. The only problem is that this interfacial tension is not easily computed. It's not computed exactly. It's not computed like it would be for two homogeneous phases. But this interfacial tension requires some, its computation requires certain mathematical dexterity to take into account the local heterogeneities of the protein, and so that could be potentially a very involved calculation. But it's not really so difficult. It's a matter of looking at the interplay between the protein charges and the geometry of the interface. So you need to realize that when water gets into the crevices, then it's likely that it will relinquish some hydrogen bonding partnerships unless the protein can actually supply a polar group to enable water to engage with this polar group in hydrogen bonding and retain or maintain its tetrahedral arrangement of hydrogen bonds. So in essence, you would say that what is really the culprit of this interfacial tension are the poorly wrapped backbone hydrogen bonds, which are called the dihydrons. So the protein essentially tries to minimize the number of dihydrons as it falls. So I am hopeful that this principle of harmonization or harmony between the protein structure and the episteric perturbation of the water matrix that it brings about will be promising and enable us to solve the protein folding problem once and for all. I have managed to recently fold from first principles a protein domain of 60 amino acids in length, and the results are uncannily close to the crystal structure. So that, of course, the computation is still very involved because we need to know the extent of frustration of each water molecule surrounding the protein. But we have a means of computing that, and so that's what's important. And so the next stages will involve faster algorithms for trying to minimize the interfacial tension and the interfacial free energy, which is the extensive quantity. And once that is achieved, I am confident that the protein folding problem will be fully tackled, and that would be a source of tremendous joy for people that have seen it as the holy grail of modern molecular biophysics. So, with that note, that optimistic note, I would like to end this lecture and just entertain some questions, if there are any. Thank you for your kind attention.